you had a good day. At least time for me to catch up on things that are ongoing or coming up. So what we're going to do today, we're going to cover, oh, thank you.
I assume this is what you're asking for? I completely forgot.
Because I've been gone for the clip is gone.
I'm going to go crazy if I have to hold this the whole time. All right, well, let's see what I can do. The clip is gone, and I'm sure if I attach this here, it's going to fall off numerous times. So I'm not going to hold the microphone. Is this going to be loud enough? I can speak loudly. I'll try to get this corrected for next time, but I'll try to speak loudly during the class. So if you do have trouble hearing me, because sometimes I may look to the board, let me know if you didn't hear something, and I'll turn around to announce it. Okay, so where we were, today's continuation of the lecture that we had the previous week before the break, when we began discussing annelids. We got through some of the lecture on annelids, but the two-part lecture, so we should be able to cover the rest of the material on annelids in today's lecture. Wednesday, we have no lecture. Exam 2 will open up at 10 a.m. instead, so you can use that time for taking the lecture. It extends through Sunday, 5 p.m. That's to allow for everyone the experience of the labs, because otherwise it was going to be some students would have it, some students would not. The reality is many of the questions can be answered based on materials presented in the lecture. So attending lab is not as important. If you want to use the time period on Wednesday for completing the exam, you can. You'll have covered all the material. You will get more intimate knowledge of flatworms and annelids though in the lab, so you can go back to the exam and revisit it after you've seen those labs, visited those labs to see how you may answer something different. We do have drafts of questions available, the comments already from GSI, so I think we have questions ready. The exam's ready to go. One of the things I worked on during the break. Where we were, we briefly introduced the phylum and we discussed aspects of the changes, at least in terms of our understanding of what the phylum Annelida is. The inclusion of other groups that were considered previously to represent their own distinct phyla, and how relationships within the group itself were highly modified. What we're going to talk about today, we're going to look at the generalized anatomy of annelids, and then look at different feeding ecologies. And specifically, while we're looking at the feeding ecologies, keeping in mind the phylogenetic relationship of the different annelid groups, like we showed last time we met, to be able to look at the evolutionary history of dietary specializations within this group. One thing that phylogenies are useful for is for tracking changes in characteristics of the group that phylogeny represents. So we'll be using a tree and we'll be mapping characters in terms of diets onto that tree to understand a little bit about the evolution of diets within annelids. And then we'll look at reproduction life cycles of the annelids, and I think at the very end we'll cover aspects of segmentation in annelids and the importance for segmentation in terms of their mobility and locomotion. All right, so aspects of morphologies. I'm going to be referring to the generalized morphology of major groups within Annelida. First, focusing on the polychaetes, those worms that have many seeding. And look at close-up view of the front end of the worm, there are two distinct body regions. The stomium, peristomium. This is the region in front. Front of the mouth. It often bears sensory organs, pouch, tentacles, and so forth. And then the peristomium, peri around, pro first. It's the region around the mouth. The area that bears the mouth. And notice the language that I'm using. I'm following what the book follows in terms of saying that these do not represent true segments. Why? Because a true segment, at least the way our book recognizes a true segment, is a segment that contains a distinct set of features. Now there can be variation from feature to feature, but at least our book takes the interpretation that these are different body regions than the segment. I'll talk later about contrary view in terms of that these are originally segments, but our book at least describes these as not being true segments. Following these two body regions, then you do have the rest of the segments that comprise the body of the worm, going down to the tail end, in which you have another region, the pygidium, the region at the posterior of the body of the worm that often bears cirri, little extensions from it, as well as the anus. Again, presumed not to represent a true segment. Many polychaetes, especially those that are predatory, have a pharynx that could be averted out of the mouth. So it's held within and then can be averted out. And it's used in feeding. Many of the predatory polychaetes have strong chitinous jaws, muscles that can be used for grabbing onto prey. We should have some of these in lab later this week, so you can see what those jaws look like. A cross-section of a segment of a polychaete bears unique features that most polychaetes exhibit. These appendages known as parapodia. They may be vascularized and serve for gas exchange. They are often, based on the name of these worms, polychaetes, they often hold numerous hair-like structure. That are referred to as setae, singular. Internally, they may be supported by stronger structures that are hair-like, that are known as acicular. Singular, in this case, this is confusing because this is plural, and then it's singular for acicular is an aciculum, kind of like datum, data is plural, data, singular, CD, and ceta, and then the larva, and larvae. These are confusing terms that we continue to utilize, but that's the way it is. But these can be large supporting structures. Not all polychaetes have them, but many groups do. And these are supporting structures that allow for movement. These are muscles that occur within the peripodium, and these are used for movements, swimming in some cases. Otherwise, internally, what we have is an intestine, dorsal and ventral blood vessels, a ventral nerve cord, and then a coelomic cavity that is encapsulated within each segment. There's a peritoneum bounding that, so you have two coelomic compartments per each segment. There are, we'll see this in close-up detail, but there are excretory organs, nephridia, that are roughly analogous to our kidneys. They're filtering coelomic fluid and resorbing water and salts. One thing about polychaetes is that they tend not to have distinct gonadal tissue. The gonad forms typically from a swelling of this peritoneum, and then gametes are produced from those structures. And so gametes are produced in segments, generally. Sometimes certain segments may be involved with gamete production. We'll see that later when we talk about reproduction of polychaetes. But in general, any segment is capable of producing gametes through this swelling of the peritoneum. There are, associated with the peripodia, these dorsal and ventral cirri, these extensions from the peripodia that can be subsequently modified in a variety of different groups. We've seen some of these pictures already. Here's an onuphid where the cirri of the peripodia are modified into these little feather-like structures. These are branchiae. These are gills that are used for respiration. Phyllodocid has leaf-like cirri. And these scale worms, the cirri, are modified into scale-like structures. And actually these, when these reproduce, the embryos are held underneath these scales. The scales are known as elytrae, for singular worms, and elytra. These can be quite gorgeous little worms, and these can be luminescent. So this is what a segment, internal anatomy, a segment of polychaete looks like. This is the internal anatomy of an oligocete. We talked a little bit about this last week, but just to reiterate, what are some of the major features that are different between a oligocete and a polychaete? I should say, these are all with quotes, these are paraphyletic assemblies. But how morphology of an oligocete differ from that of a polychaete? What are some of the major features that you can see that differ between these slides, or these images? No differences?
Yeah.
The Parapodia and polychaetes.
The Parapodia and the polychaetes, exactly. So the major difference is, if this was an oligochaete, we wouldn't see the side extensions, the appendages of Parapodia. Very obvious difference that we will see when we examine different oligochaetes and polychaetes in the lab later this week. But it's a predominant feature that varies. Should say that, again, these are generalized anatomies of oligochaetes and polychaetes. There are some polychaetes where parapodia are much reduced. Many tube-dwelling polychaetes have very reduced parapodia in comparison to those that are errant, free-living, that use these for movement. The other features that differ. Obviously, the musculature associated with the parapodia, but also based on just simply the names, polychaete versus oligochaete. These are the CD. Those are the CD on the oligochaete. Again, oligo means few, so they have very few CD present. Other major feature that differs within the oligochaetes is that their digestive tract is differentiated as you go down along through it. They have different regions of the digestive tract where if we did dissection of a polychaete, which I have done in past labs, so we're not going to do it this year just because the polychaete internal anatomy is rather boring. There's not a lot of differentiation throughout it. Whereas with the oligochaete, you see considerable differentiation into particular regions of the digestive tract, into a crop where food material is stored, a gizzard where food material is ground up, and then the remainder of the tract, which is very similar in appearance to that of the polychaete. But oligochaetes have some distinct features of their gut throughout them, whereas polychaetes typically do not. I mentioned polychaete gametes, typically formed by these swellings of the peritoneum. I did mention polychaetes tend to be dioecious. So you have male polychaetes and female polychaetes. Within this internal anatomy, we have seminal vesicles, testes, ovary. Obviously, this is a hermaphroditic individual that is capable of producing both male and female gametes. I lost my eraser. Oligochaetes and leeches, which is all part of the large clade Annelida. All of these individuals, all of these species, are monoecious or hermaphroditic. They have both male and female gonads. What's also different with the clitoris, they have distinct gonads that are produced in particular segments. So when we talk about reproduction, we'll see how which segments those occur in vary from one group, one species, to the next. Looking at another cross-section of the book, this shows similar internal anatomy to what we observed for the polychaete, except for the lack of the parapodia, the presence of
intestine, blood vessels, ventral nerve cord, peritoneum, lining, coelomic area, nephridia, structures that involve an excretion. Something I didn't mention, we'll talk about again, we talked about the reproduction of fiddlets, is the presence of a patellum. Secretes mucus, which ultimately becomes the cocoon from which embryos develop. Okay. Probably all seen, if we've examined an earthworm, they have a swollen region, picking segments, that's a patellum. Ever observed the leech? They're more difficult to observe because they're typically only expressed when the animal is reproductive. Nephridium. Again, it's this tube funnel that opens into the coelomic cavity of the segment in front of where the structure occurs. So it's drawing in coelomic fluid from the anterior segment. The ciliary action draws fluid in. It comes into a vascularized network. They're very analogous to how things work in our kidneys, to where salts and water are absorbed from them, and then the dilute urine is expelled through the nephridial pore that opens externally on each segment. Common in most animals. Leeches, there are a number of segments externally, but internally they have no septal separations for the most part. They too are hermaphroditic. You have testes, you have ovaries, complete reproductive tract, some differentiation of that reproductive tract as you extend down the length of the intestine. Suckers, anterior sucker, the mouth, the proboscis that could extend in for feeding. Many leeches engage in blood feeding. So the proboscis, mouth, they also express an anticoagulant to be able to continue feeding on blood from their host. Now the ceti, what give oligochaetes and polychaetes their name, are, again, few in oligochaetes. We're going to be examining earthworms this week in class. In lab, we'll be able to see the paired sets of ceti that emerge from each segment. It varies, different types of oligochaetes. This one has larger number of ceti emerging from each segment. There are echeuran worms that have, like the name implies, echeura, spines at the tail end that are quite noticeable. They often do also possess these ceti posterior at the anterior end as well. Polychaetes, they're much more prevalent, obviously, there are many of them associated with them. Again, ceti, merge, fascicula, supporting the parapodia, and then these are used to aid in movement. They exhibit a variety of shapes and sizes. Some of them, like from this tube-dwelling polychaete, are relatively simple and hooked, presumably used for staying within a particular region of the tube. Others may be compound, to where they consist of numerous separate parts and they articulate. As shown for this worm here, with these articulations that they're able to rotate. In terms of the biomechanics of how that's associated with different movements, I'm not completely aware on what the adaptations of an articulated versus an unarticulated zeta within polyhedrons is. But there are some polyhedrons that have numerous elongate ends. These are frankly quite gorgeous. These are fireworms I was talking about 2 weeks ago, I guess. Last lecture. Long seedings that are presumably used to deter predators from feeding on the fireworms. Fireworms themselves tend to be predatory on various types of magnetics, soft corals and hard corals. They have these very long seeding. And I think what I mentioned last time was our book, and it's kind of just this latest edition of the book, comments on the fact that it was previously thought that these CD were hollow and were used to pass venom to the prey. But our book says that's been disproven, that's shown not to be true. And there was a paper that came out a few years back that did suggest that in certain species of fireworms that isn't the case. But some other more recent studies looking at CD, there's a clear region in the CD, at least of this hermaphrodites. This fireworm has what appears to be a hollow region to it, a little tip of fluid at the end. They think it can be serrated. The serration makes sense. It kind of goes in, it's going to be tough to get out. So that would be physical pain, that would deter predation, venom, which again, if you pick up a fireworm, it burns like a fire. So there is some noxious component present to it. But at least this study demonstrated that they may be, for some species, they may be hollow. Another paper that came out more recently looked at tissue associated with the parapodia and evaluated whether or not there's any toxic, like peptides, that are being expressed in that tissue. We can take a particular tissue and we could get all the messenger RNAs from that tissue. Those messenger RNAs later would be translated into proteins, right? So you could look at which messenger RNAs are being expressed, and then you could attempt to identify those messenger RNAs. They found what this particular study did was they looked to see if any of those transcripts, those messenger RNAs, would encode toxins that we find in other venomous animals. They found a lot of the CLECs, which are C-type lectins. These are common toxins that you find in various snakes. Looking at all, they looked at a variety of different things. They found 13 total spider toxins. So transcripts that would produce proteins or peptides that resemble toxins from spiders. Kunitz is another one that you find in snakes. And then there are even some conopressins. These are conopressins because they are structures, they're peptides that are found in cone snails. So there's a number of different transcripts that at least resemble toxins that you find expressed in other venomous animals. So it's not proof that they are actually producing a venom, but it's suggestive of they are. So, need for further validation. Their title are fireworms venomous.
They don't have the definite answer, but there's at least some suggestion that they're producing toxins. And they're being potentially transmitted through the CD. It's also possible the CD are not all hollow in all cases, and it's just a rupture of skin and associated tissues that may be expressing these toxic substances. Those are getting into potential predator. Annelids, we didn't discuss too much with regards to sensory structures of chordates, but we certainly could have featured much of the lectures on vertebrates in terms of the evolutionary sensory features. But we'll spend some time focusing on annelids because there are a lot of different sensory features that have evolved within annelids that are shared by a variety of other groups, but some that are completely unique to annelids. So annelids do have eyes. We'll see this in at least preserved leeches. I'm not sure if the living leech that we have has well-developed eyes or not. Not that they're well-developed, they're just little eye spots. But leeches often have little eye spots on their interior. There are some polychaetes that have rather large, in this case red, eyes. Probably one of the largest eyes in terms of body size for a polychaete. There are polychaetes that bear eyes on their radials, their feeding appendages that emerge from ostomium. See the little eye spots present on the branchia, fossa of them. There are distinct tentacles too that specifically have eyes that develop within them, and a variety of other beasts that have eyes associated with other regions. There are some that have eyes associated with the pygidium. So eyes may occur on lots of different parts of the worm, mostly concentrated in the head, but they can occur throughout the different parts of the worm. Some of the image-forming eyes that we find in certain polychaetes, the development's been studied, and they may consist of pigment cells, and photoreceptor cells that later form a complex structure. The pigment cells also contributing to the formation of a lens. So two different cell types making up an image-forming eye of certain polychaetes. Others, as we can imagine, there are sensory cells that are associated with palps and tentacles. And so we have these little batches of ciliated cells associated with various structures that are chemosensory. One feature, well, if memory serves me correctly, it was noted on our text tree of Annelida as the nuchal organ being a distinct feature of annelids. These are chemosensory. They're typically paired organs. That are observed as these concentrated regions where cilia occurred, ciliated areas. On the prostomium, we may have the opportunity to observe these in some of the samples that we have in lab this week. But if not, there are some different views
of the nuchal organs, very large organs. They can be concentrated in certain pits on the head. See here, different regions of the head. So these are usually areas of highly concentrated ciliated cells that are engaged in chemosensation. And then the other, we saw these with Nigerians, statocysts. Balance organ. Typically you have these ciliated cells, nervous support, and little statoconia, little grains of sand, grains of some mineral, that through their movement it's monitored so that the individual can keep track of its orientation and balance. So I had originally planned to talk about segmentation here. So if you have slides in your hand and you're looking at them, or on your computer and you're looking at them, I'm going to reserve talking about metamerics until the end of the lecture. I want to talk now about feeding. So you may have to jump a few slides ahead if you're looking. Can I give a jump a few slides ahead? Annelids exhibit a variety of different feeding modes. I find the evolution of feeding ecology fascinating, because what it does is any changes in aspects of feeding ecology opens up new exploitable habitats for individuals. So evolution of diet is an important way by which species can diversify into a number of different habitats and are not competing with closely related types. So it's an interest of mine. Within my snails, I study my snails because of their feeding specialization, but for God's sakes, they're all predatory species. They just differ in aspects of what they're eating. Annelids, on the other hand, exhibit a variety of different feeding modes that we'll talk about. Deposit feeding is a mode of life that lots of different animals employ. We talked about sea cucumbers as being deposit feeders. Anyone can describe what deposit feeding is? Feeding on deposits. What is a deposit feeder? How does a sea cucumber feed? I don't think we saw any in the lab feeding. They usually don't cooperate in that way, but we saw some videos. How do sea cucumbers feed? It wasn't that long ago. No one remembers that. Sea cucumbers feed. They're modified tube feet, use this tentacles, and what are they doing with those tentacles to get food? We even saw a video of a sea cucumber pooping, right? Anyone remember what it was pooping? Nobody's going to volunteer. Yeah. Sand. They're feeding on sand. I think it was Raising Arizona. A movie with Nicolas Cage, where there's a feature inside the prison at the beginning of the movie. And the guy was talking about how difficult it was getting food for his family. And then he said that he ate sand. And I think it was supposed to be funny. Sand, you're not going to get any nutrients from sand, are you? It's a mineral. You're not going to get anything directly from sand. But sea cucumbers eat sand. What are they getting? Where are they getting the nutrients from? The organic matter that's associated with those sedimentary particles, right? So deposit feeding is ingesting some material and then getting the nutrients from that organic material associated with that sand. So it's organic material associated with some kind of sediments. It could be clay, it could be mud, silt, sand. So that's a feeding mode that a number of different annelids exhibit. Second, that we'll touch on, only a few groups have evolved this particular mode, but using a mucus net to feed. So essentially producing mucus and then filtering water through it and then consuming the net, which contains all the organic material that stuck to it as the water was filtering through it. A few different groups show this. Suspension feeding also referred to as filter feeding. They're feeding on material that's in suspension, material in the water. Who we talked about already. Crinoids are suspension feeders, filter feeders. Some sea cucumbers, talked about, they have tentacles that are adapted for, and they put them in the water column, and they trap little particles, or they latch onto little particles, and those are brought into the mouth. So suspension feeding is something used by a variety of different groups. Here, they're using some kind of specialized structure for capturing particles from the water. Predation, I don't think we need to describe. It's feeding on other animals. Blood meals, I don't think we need to describe. Parasitism, I guess a type of predation where the parasite is not killing the host. One of the others, we'll talk more about this when we get to the examples, but there are a number of different polychaetes, predominantly members of one group that has symbiosis with various bacteria. Most of them have symbiosis with chemoautotrophic bacteria. You saw the video before today's lecture that kind of focused on some of these worms that have these symbiosis with chemoautotrophs. These are different means by which worms get nutrients. Generally, there's some variation, and when I'm talking about particular groups, there are certainly some exceptions. Most predatory species predate on some other animal, but not always. Some may feed on algae occasionally and other things, so they're not strictly predatory. I think many of these that have symbioses with chemoautotrophs are strictly relying on the chemoautotrophic bacteria. They cannot switch to a mode by which they're consuming other things. Many of these have lost their guts. They're incapable of feeding on their own without that symbiotic association. So I just want to use this to say, this is not specific to everything as I'm going through these examples. Do you have a question? No?
So you remember, you don't remember, but this is a tree we talked about at the end of the last lecture. And this, again, having a phylogeny in hand allows you to evaluate how different features of the critters within this phylogeny have evolved. So we have a big list of different feeding modes. I already said deposit feeding is exhibited by many different annelids. Good bet is that deposit feeding represents the ancestral feeding mode for annelids. I'll walk you through why that may be. So let's, before we do this, first look at this tree, and what we have are these two families that are the most basal compared to the rest of the annelids. So one question is, well, what do these eat? Megalonids, Oenids, have these little tentacles with these palps that are used for deposit feeding. These little worms, much like a sea cucumber, will use those tentacles to latch onto particles of sediment, and they consume those sediments. The Megalonids will use these palps to do the same, to draw in sediments into the mouth of the worm. So these engage in deposit feeding. So we're mapping that onto the tree. Both of these, at least most members of both of these groups, tend to be deposit feeders. So presumably ancestrally, this group was a deposit feeding worm. Not much has really changed. Again, there may be exceptions within both of these families, but in general, they tend to be deposit feeders. Earthworms. I think this is one you easily recognize as a deposit feeder. What is it doing? It's eating dirt, soil all day, and taking the nutrients out and moving out the sediments. Very important in gardens and so forth, because it's moving soil and so forth. Many oligochaetes are deposit feeders. Oligochaetes are part of Clitolata, which is up here. Clitolata also includes leeches, so we know that leeches are deposit feeders. So this isn't true for all of them, but at least for now, members of Clitolata are deposit feeders. Yeah, question?
Okay, so how are earthworms like technically good if they eat up the nutrients?
They're eating the nutrients. Good point. So they're eating the nutrients from the soil, but they're aerating the soil. They're moving soil up from bottom layers and bringing it up to higher levels. And they're not, I guess they're not completely removing everything from the soil. They're not making it a sterile soil, but they're aerating the soil and moving it around. Good question. Clitolets, these guys, I don't know what the name is for those, that clade. Terabellids, this is a figure from the text. Terabellids have these long tentacles that are used in feeding. They're bringing sediment material to the mouth. These also are deposit feeders. Pelobelliformia is up here. Just one more deposit feeder, I think. The lugworm, beautiful lugworm that lives in a tube. And we can see, this is a polychaete, but it has reduced parapodia. There's an example of a tube-dwelling polychaete with reduced parapodia. These live in a U-shaped burrow where sand is brought in through one end, sand is constantly falling down, and they are consuming the sand. And then eventually, coming out the tail end, they're moving, they're able to move water through peristaltic tractions of the body into here, which allows their sand to continually fall out. Another group that's deposit feeding. Ranicolidae. Here, we could walk through this further, but I'm not going to do that to you all. Many of the groups here, many of the groups in this clade, Sedentaria, are deposit feeders. Not all, we'll see the exceptions. But many are. So what we can do is say, okay, well, ancestrally for the group, Sedentaria, they were deposit feeders. So we have these two that were deposit feeders, that group that was a deposit feeder. This group
for the most part. We don't know all for sure. But most of these families are deposit-feeding worms. Sipunculates, one of those groups that was a phyla by itself, now recognized as an annelid. They're also deposit feeders. So deposit feeders are throughout this tree. So I would use that as evidence to suggest that ancestrally, annelids were deposit feeders. So the ancestral feeding mode for annelids was deposit feeding. And that's from where all the other feeding modes arose. So the next question can be, well, for those other feeding modes, how many times did they evolve? Is diet rather labile trait? Can it change very easily? Or is it rather conservative? It changes just a few times. So we'll see that by mapping out the other characteristics on this tree. So instead of just having DF on particular branches, we'll make the assumption that DF is the ancestral feeding mode. It's an assumption. It's probably the simplest way to explain where deposit feeding falls out on this tree. Okay, so mucus net feeding. We talked about the parchment tube worms last week briefly. We'll be able to see some in class, hopefully some live ones in the lab later this week. These feed again by producing mucus associated with particular segments. They live in a U-shaped burrow. They have particular modified segments, referred to as these fan regions, that are moving water through. So material comes through, water comes through. Any particles are attached onto the mucus net as the water is passing through, and then ultimately the animal consumes the mucus net and gets all the nutrients from those particles that got embedded in the mucus. Catopterids. Not all, but some echidnas do something very similar. This is an innkeeper worm, known for an innkeeper worm because there's often a variety of different commensal organisms that are living in the tube with them, or her. These, they don't do this in a glass tube in nature, but in a lab, in a glass tube, they'll secrete a mucus net, and it's getting darker red because they've stained particles in the water red, and so these are different periods of time. Through peristaltic contractions of the body, water is brought through the tube, and then ultimately the animal consumes the mucus. So these are the echidnas. Echidna occurs here, and again, this is one of the cases where there's, the innkeeper worm is one that uses mucus net feeding, but most echidnas are deposit feeding. But this at least is one place where mucus net feeding evolved. Catopterids are another. So in terms of mucus net feeding, this is something that has evolved at least twice within the group. Do I have it here? Oh yeah, I put it on. Most nereids, and hopefully we have some nereids in the lab too, most nereids are predatory. But some will engage in a mucus net feeding strategy. So it's something that appears to evolve multiple times with the animals. Suspension feeding. Filtering material from suspension, the water. There are Christmas tree worms, which are serpulid polychaetes, phyrorbid worms, which are serpulid polychaetes. Hopefully we have these in the lab. These are really cool. And then there are feather duster worms that have these radials for feeding. And we have little eyes associated with the radials, and they're brucilia on the little pinnules that emerge from each radial. They're capturing particles from the water column, bringing those particles to the mouth for feeding. We go to our tree, and all the groups that are suspension feeders are more closely related to each other. All of the families. So here the sabellids that we saw, serpulids, sabellarids, we should have in lab this week. And the other groups, trapeziids, spionids, most of these are also suspension feeders. There are sabellids that will occasionally deposit feed, use the tentacles for feeding, but for the most part, this is the clade, and the only clade where suspension feeding is involved. Mucosat feeding, multiple times. Suspension feeding only once. Possibly due to the fact that it required evolution of distinct structures that were used for feeding. So it suggests that there's homology between the tentacles of serpulids and radials of sabellids. Only once suspension feeding involved. Predatory gastropods, predatory polypes. They are numerous. Some of these have this is a scale worm, the beautiful jaws of the scale worm. Here it is over here. Many have very large proboscis with jaws associated with them that are used for capturing prey. Some jaws have venom glands at their base, so there are venoms that are produced that also aid when we capture prey. We go to the tree, and except for an occasional myriad that engages in mucosat feeding, all members of this group are predatory. The predaceous feeding mode in annelids evolved once, the common ancestor of all of these families, and once also in the fireworms that we talked about earlier. Fireworms which tend to be predatory. Again, most of these tend to be predatory. They're not obligate predatory species. They will occasionally feed on other substances as well, algae. Yes?
It makes sense that the predatories are all in the non-sedentary category, because how can something be predatory and not move? Or I mean, it's possible, but it's more likely for it to emerge in the works that can move.
Right, and I didn't show the video. I know I got the slide here, and there is a video of the bobbit worm, which is it's actually an errant polychaete that does live in a tube, and it sits at the bottom of the seafloor. If you have that big picture, there's a big picture of a horrible looking jaw in the slides that I put out. It does sit in the bottom in the tube and waits for something, sometimes little fish that he'll latch on to. But exactly right. And that's probably, which came first? Errant feeding, errant, I mean, all these are errant. They also all happen to be predatory. You probably can't have, we do not have strictly tube-dwelling sedentary polychaetes that are predatory. Unless I came up with an exception. I can't think of any exceptions off the top of my head, but don't think any of these are actively preying upon other animals. Yeah, errant and predatory go together. Amphinomids are errant. In the olden days, when we talk about errant polychaetes as a lifestyle, amphinomids fit that bill. So too would go with Magellonids and Linnaeus. But there are some that are still deposit feeding. This group here is all deposit feeding. Blood meals, leeches, these are clitoris. Most leeches engage in hematophagy, taking blood from their hosts, and there's even leeches on snails and various other types of vertebrates. There are terrestrial leeches and there are aquatic leeches. So this one isn't all that exciting because there's just one group within the clitoliths that engages in blood meals. Parasitism. Most worm groups we think of, flatworms for example, are chock full of parasites. Annelids nematodes, we get to nematodes, my God, lots of parasitic nematodes. But annelids, this mode of feeding is really the exception. There are very few annelids that are true parasites. One group that does have parasitic life cycle, the mesostomids. Meso means to suck. Stoma, obviously mouth, the sucking mouth. These are odd little annelids that previously weren't even recognized as annelids. They were initially thought to be flatliners or some other type of animal because they did not exhibit features that were obvious as annelids. Many do have little CD, hook-like CD, that they use for attaching on the host. But many of them too are just commensals on, anyone recognize who these are? Some crinoids. Here's one of the mesostomes that is commensal on this. So not really a true ectoparasite, it's just getting additional food material that's brought down here, it's feeding on the food of the crinoids, stealing its food. Kleptophage. There's another one here. They exhibit a variety of different color patterns and so forth that look like they're mimicking their host. So these aren't true parasites, they're stealing food material. But there are some mesostomes that occur in internal structures, largely of crinoids, but some asteroids and some ophiuroids, various chondromes. And these then are feeding on the host itself. And so these are, some mesostomes are parasitic. Go back to the tree, that's just one branch where that particular feeding mode. The last major one are the symbioses. And these are in a group of worms we talked about last lecture, the babuinids. Offerings, beard bearers, vestiferans, vestment bearing, that have no gut. At least in adult stage. In larval stages, they do tend to possess digestive tract. But as in the adult stage, no digestive tract. So the coelom is filled with cells that comprise the trophosome that is rifled with full of hemoautotrophic bacteria. Many of these are associated with hydrothermal vents, as these Riftia species are here. There's lots of chemicals being produced at the hydrothermal vents, and so they're using, or at least the bacteria in them are using chemical energy carbon. Right? Plants do this through photosynthesis, through light energy. Chemoautotrophic bacteria can produce organic compounds through chemical energy. In the movie before, some of these can be quite large worms. They tend to occur at hydrothermal vents. You were to take one, we don't have any in the lab, they just be gross. But if you cut open its main body, you see the trophosome within, and the trophosome is the area that contains cells that are rich with symbiotic bacteria, chemoelectrophic bacteria. So what is happening is the worm, and worms, this plume area where a lot of the gas exchange takes place, there's a lot of hemoglobin present there, that's why they're so red. It's taking in hydrogen sulfides, oxygen, and then bacteria, and using that to convert CO2 to organic molecules, larger molecules that they take part of. There are a variety of those that are associated, the siboglinids that are associated with hydrothermal vents. There are a number of other that tend to be more slower developing siboglinids that occur in cold seats, and there's some that occur with whale falls, sources of chemical energy. One that isn't a true chemoautotrope, but it is a siboglinid, are the bone worms. Ocedax, again, the movie at the beginning showed some of these bone worms. These are fascinating worms that colonize whale falls. So whale dies, the carcass falls to the surface, and then it's a rich source of nutrients for a variety of different organisms. And even the video showed a bunch of bone worms as well as brittle stars that were around the whale fall. And these bone worms colonize the bone and are absorbing fats that are present within that bone tissue. And then there are symbiotic bacteria that digest the fats. So they still have a symbiosis, they're siboglinids, and they have a symbiosis, but not with the chemoelectrophic bacteria, but they do have symbiosis with other bacteria. Yeah, question? What's that? I didn't hear you. Rest after set up the rest of the interpreter? Oh. I assume after rest of interpreter. This is something I didn't erase. Sorry about that. But these two are an interesting beast because all of these worms that you can see coating this whale, as well as all of these here in this close-up, are all females. Most polychaetes are dioecious, males and females. What are the males in this case? The males are in the oviduct of the female. If you want to be if you want to be successful in fertilizing the eggs of the female, hang out the oviduct, like these males are doing. Here are the individual males in close-up that are occurring within the female oviduct. So these larvae settle on a female, and then eventually find their way to the oviduct. So it's a case of dwarf males. A lot of deep-sea fishes also exhibit this feature, where the males are small and they latch onto the female. And it's something common in deep-sea organisms, suggesting that finding a mate in the deep sea can be difficult. So if you find one, you latch on, or you find a place to settle into, so that both are successful in reproducing, females and males. If we go back to this, where symbioses tend to occur, again, there are exceptions. There are some other polychaetes that engage in symbioses, but siboglinids are one of the families that exhibit this feature exclusively. So if we take all of this, we observed a few different things, right? Predatory feeding modes evolved within the errant polychaetes and amphinomids, just a couple of times evolved. There haven't been a lot of transitions within this mode. Suspension feeding, also something that appears to evolve. So there's a bit of dynamicism to the evolution of feeding ecologies within the annelids, but fairly conservative. Right? We don't have, alternatively, what if this is a predatory group, this is a predatory group, this is a predatory group, and so forth, then we would be able to conclude that evolution of predatory feeding mode, something that is very labile, something that's easy to do. Doesn't appear that way. Yeah, question? Aren't there predatory? There are some that are predatory leeches that will feed on NRC. That's why I said those are, 75% feed on blood meals, so yeah, we can put some leeches that are predatory feed on their worms and their animals. So, not everything is perfect. This is a generalized view of the evolution of different feeding groups. Yeah.
I might not know enough about leeches, but why are leeches considered sedentary animals? Is it just where they are in the tree?
Yeah, exactly. That's a clade. In the past, sedentaria or sedentary polychaetes was a reference to certain types of polychaetes. And so they're now just happen to fall out within that clade. But exactly right, they're not, I wouldn't describe them as a sedentary lifestyle here. All right. We don't have much time to talk about growth and reproduction, but there are a few interesting features that annelids exhibit. Most annelids tend to grow along the posterior, so new segments are added as they continue to get longer. But there are some interesting regenerative abilities that various annelids now don't go kind of cut in half an earthworm, I think you did too. But some annelids, if they're accidentally amputated, they will, the front end will grow tail segments, and the tail end will grow front segments. Certain groups, not all groups. Others may engage in spontaneous fission, architomy shown here, where the different parts will grow their missing parts. And then a bizarre one is this notion of peritomy, where certain segments in the worm will begin to develop tail and head segments, but will remain attached for a period of time. Psyllids are one that do this, and this is just such a gorgeous image of a psyllid that's in the process of doing this. So the further back you are on the tail, the more developed those segments are. So ultimately, this individual can butt off. So there are some worms that engage in asexual reproduction. Oh, and this is a weird one. These are some psyllids that will just produce multiple ones all attached to the tail, and they start growing in one segment, and another one starts, another one, and so forth, where you get this bizarre looking worm. But otherwise, annelids are like the other groups we talked about, where most of the polychaetes are dioecious, producing male and female gametes, then fertilize to produce embryo, and then annelids have trophophore larvae that will metamorphose into an adult worm. This is a figure I showed long ago. We talked about larval ecology. There are some that will develop directly from a fertilized embryo, or that will go through a yolk type embryo. So there's a lot of variation in reproduction with the polychaetes. Oh, I have to put a little video. There are some polychaetes where posterior segments of the worm are those that will produce gametes. So this is either a male or female, and the atope is the front part of the worm, where there's no gamete production. The epitope is the part where gametes are being formed within the segments, male or female. And in some species, the epitope breaks off
and just goes swimming on its own into the water column. In others, it's a whole worm that swims around. In the South Pacific, we did show just a little bit of this. South Pacific, during certain times of year, these emerge. It's usually like in November, after a certain tide. And there we get the scene underneath the water. These are tons of these epitopes swimming. And in the South Pacific, it's a delicacy. So there are people in the water collecting these because you get a good batch. I know flavored fish pate, Pololo flavored fish pate. I know in American Samoa, people will eat them straight out of the water, or less traditional meal, they'll scramble them, scramble eggs. So it's actually crappy. I guess you've got sperm and eggs with the chicken eggs. It's got a fishy taste to it. Interesting. Cultural phenomenon in the South Pacific. The trophophore larvae of most polychaetes is a generalized image of what one looks like, these little bands of cilia, and they tend to vary from one group to the next. Cycunculids have a trophophore larval stage, but they also have later stages that are subsequently distinct. That's for most polychaetes, generalized reproduction of most polychaetes. Oligochaetes are very different. Oligochaetes are hermaphroditic. Oligochaetes engage in copulation. Individuals tend to cross-fertilize, so they don't self-fertilize themselves. They will line up against each other in different orientations. There are sperm ducts and ovaries associated with particular segments. The patella of these two individuals are producing mucus, so you get this mucus secretions. Fertilization of oligochaetes takes place within the mucus. This mucus ends up forming a cocoon that is released that contains the fertilized eggs or the developing embryos. Within leeches, it ends up being the same. Leeches will produce a cocoon with developing embryos, but the leeches will line up next to each other, and actually sperm is transmitted into the other individual's oviduct where internal fertilization takes place. So within clitoliths, they're all hermaphroditic, but oligochaetes, certainly, their kin, have external fertilization. Leeches engage in internal fertilization, and then the embryos are released into a cocoon of eggs. And I mentioned this earlier. With oligochaetes, in some cases you have adjacent segments that bear the testes and the ovaries. In other cases, they're vastly separate from each other, and they occur in different segments. So oftentimes identifying oligochaetes is looking to see which segment it occurs in. Testes in segment 5 here, segment 10 there, segment 11 here. I do not have a chance to talk about segmentation. We'll see if maybe we talk about it a little bit in lab on Thursday, Friday. All right? So I'll have office hours today from 2 to 3 is normal. I'll have office hours again 2 to 3 on Wednesday. If you're taking the exam and you say, what the hell does he mean with this question? Send me an email or visit me at office hours if I happen to be using office hours during that time. Let me know if the question isn't clear. We try to make sure the questions are clear, but we're not always perfect.